Hello, everyone, and thank you so much for joining me today. We are going to look into Module 4 in the Common Technical Document or CTD, which is used for several types of submissions in all of the countries where we are using ICH guidelines. So here we are looking into again the content of module 4. You know very well that in CTD backbone we do have 5 modules that you can see on the top: module 1, 2, 3, 4, and 5. We already looked into module 1, which is for the administration, and here I'm just talking to you, showing you the module I would say 1 for Canada. This is where we do have again the table of content, we have application information, electronic review, etc. So I will bypass that since it has already been discussed in previous lectures. Then we have module 2, which is related to I would say the overall information or summaries of module 3, 4, and 5. And of course you can see the structure of the backbone for CTD. This is as per ICH guideline M4. And then we also looked into module 3 based on again ICH M4 guideline, where I showed you all of the different components in each of these different modules. And then we looked into of course the content of those files as well. So today we're going to look into module 4 of the CTD or Common Technical Document as per ICH M4. So module 2 through module 5 are universal. It means that if you are doing an IND, an investigational new drug for FDA, if you are preparing a new drug application for FDA, if you are using again this I would say the CTD for a new drug submission for Health Canada, or for a marketing authorization application MAA for Europe, or for the application of clinical trial application CTA in Canada or in Europe, CTX or critical trial exemption. It can be for a drug master file submission. So all of these modules 2 through 5 are similar. So module 1 is regional. It will depend on the type of your submissions, and of course in which country you are going to submit your application. So let's go to the again module 4. Of course here you can see again different internships that are offered by Crown College of Canada. And here, of course, I chose regulatory affairs for the new drug submission in CTD format. And we are looking into the examples of files to use if you would like to, again, create the same documents for another drug. So here, first I'm going to go over the table of content for module 4. This is where we are going to start giving information to scientific reviewers and officers in regulatory authorities in different countries. And this is related to animal studies. As you see, we do have the study reports, where in each folder or subfolder, we can call them modules and submodules, we can add all of the study reports. For example, when you are in module 4, submodule 2, submodule 1, we are looking into the pharmacology. So we will have to prepare information about the studies that we have done in animals, in different animal models as well, related to the pharmacology. So when we would like to add information into different submodules of the CTD, we are going now to add all of the safety, or we say the study reports related to the study of primary pharmacodynamics in module 4211. This means that we will have, for example, 20 studies in different animal models, And we do have several study reports. So study report 1, 2, study report 3, 4, 5, up to study report 20. And we are going to add all of those study reports in a module 4211. Obviously, we can also add all of those again study reports in one document as well. This is why you may see in different submissions, or we are having 20 files, PDF files in each of these modules, 4211, 4212, etc. So when the reviewer is going to open that folder, he or she is going to be able to look into the study reports related to pharmacology. If not, we can again, as I mentioned, we can add all of the study reports in one file, then create a PDF, and then obviously to add bookmarks on the left side of the PDF. In this case, the reviewer will still have access to all of the safety reports and will be able to review your application. Now, what goes into, again, as module 4211? Here we're going to add primary pharmacodynamics. But what is primary pharmacodynamics? Pharmacodynamics is about what the drug does to the body. So we're going to look into the study of the drug on the different effectors, on different, I would say, intracellular pathways, on different channels, on different maybe G proteins, for example. Anyways, we're going to look into the effect of the drug, and this is what we call primary pharmacodynamics, because this information is directly related to the therapeutic use or the future therapeutic use of our drug. So what we call secondary pharmacodynamics. The secondary pharmacodynamics and any study that is related to this field are going to be added into module 4212, as we can see here. And this is related to any discovery that we did or any study that we did on pharmacodynamics that are not related directly to the future therapeutic use of our drug, but still we add them here. Now you may say, okay, but we don't have any secondary pharmacodynamics study. This is okay. This submodule is optional. So if you don't have any study related to the secondary pharmacodynamics, just leave this module blank, empty. Okay, well, of course we don't remove any module, we leave them as they are in the CTD backbone, and we don't touch the structure of the CTD as per ICH M4. And we can now go to the next module, which is about the safety pharmacology. So we are looking into animal studies. We are going to look into the pharmacology. So what the drug does to the body in different animal studies, and we are going to have maybe a list of 10, 20 animal studies where we did look into the safety profile of the drug in animals. So we have been testing the drug in animals, we have been looking into what the drug does to the body, and we have been looking into different signs and symptoms of untoward medical occurrences experienced by I would say our animals. Okay, I shouldn't call the term medical, this is veterinary, but anyways, we are looking into side effects, I would say, when we are looking into the safety pharmacology. So again, I repeat that you may have 20 studies related to the pharmacology of the drug related to the safety of the product in animals. You may add several files in PDF format related to the study, again, the studies that you have done, or you can have one file where you are going to add all of your study reports. And of course, in this case, don't forget to add bookmarks in your final PDF for the information on safety pharmacology in module 4213. Then we will also look into module 4214, where we will look into the drug interactions. This means that based on our research plan, we are going to also test not only what our drug is what is going to do with the body, but we will also test at the same time the use of our drug with other existing drugs on the market, again depending on our research strategy. And then here in module 4214, we are going to add all of the reports of studies that have been done where we have been looking into the drug-drug interactions or interactions of other drugs with our drug in animal models. Then in module 422, we are going to look into the pharmacokinetics. Pharmacokinetics is about what the body does to the drug. This is where again we are going to have several animal species. We are now going to look into adding information about the analytical methods and validation reports, because we need to talk about the methods, how you have been again testing the absorption of your drug, the distribution of your drug in your animal models, the metabolization of your drug in different animal models, and of course their metabolites, and then the excretion. So it can be through the feces, urine, saliva, milk, for example, or by exhalation. So again, in each of these different modules, so 4221 through 4225, you're going to be able to add several study reports where we have been looking into absorption, distribution, metabolization, and excretion, or ADME. And again, if not, you can always put all of your study reports in one document. Don't forget again to create a PDF file and then to add bookmarks. This is how we are going to help the reviewer to be able to look into a well-organized information that we will give them. Then in module 4226, we are going again to look into the pharmacokinetics and drug interactions. Why? That's because, as you know very well in pharmacology, we also have the effect of other drugs on the effect of our drug. We may have some competition for some receptors. We may have, for example, some type of I would say interaction in between the other drugs and our drug, and this may of course affect the absorption level, the distribution, the metabolization rate, and of course the excretion as well. And of course we can look into other pharmacokinetic studies, and again here these are studies that we did in addition to what we have done, I would say, just for looking into the I would say the direct effect of the body on the drug in our body. Then in module 423, we are going to look into the toxicology, meaning that we are going to expose our animal species to doses that are not natural, again natural, I would say normal, based on our research strategy. So here we start to give a single dose to animals, and then we look into the effect, efficacy, and safety of the drug. Then we are going to reach doses that are higher, and then if they are higher, we are going to see not only effects on the efficacy of the product, but also we will see some signs and symptoms or side effects that we call overdose. Now, we can cause those signs and symptoms of side effects by a single dose, where we are going to again add all of the study reports related to the single dose toxicity studies, as well as we can repeat a dose, and in this case we are going to have repeated dose toxicity studies where we are going to add into module 4232. Don't forget that when we are going to do one single dose, we use it, we will have the increasing level of the product in the blood of the animal, and then we will see a decrease in the amount of the drug. Now, the decrease can be linear or it can be exponential. If this is linear, there is a sign that one of the indications that this is mainly, I would say, excreted by urine. But if we are looking into an exponential decay, we can now look into more complex metabolization pathways where we can now go through the hepatocytes. Again, not only the hepatocytes are going to cause, or I would say, metabolize the drug, but we also have other organs where they also have CYP 450 enzymes that are involved in the metabolization of the drug as well. And then we are going to look into genotoxicity. So looking into the genomes, this will be in module 4233, where we look into the in vitro and then in vivo studies. And we are going again to add those study reports in modules 42331 and 42332, where again you have the option to add all of the safety, study reports, one after the other one in PDF format, or we can combine all of those study reports and then to add them in one document and then to create bookmarks. Then we'll also look into the ability of the drug to cause cancer. This is where we will have studies on carcinogenicity, where we will have long-term studies, because as you know very well, cancer doesn't appear right away. It will take some time, and this is why we may have sometimes, we may go on the market, but still we are continuing to do carcinogenicity studies. And again, if we find again any indication that their drug is causing cancer in animals, it will then trigger obviously a notifiable change, and we will need now to update our product monograph in Canada, or the product label in the US, or the summary of product characteristics document in again in the Europe, or of course if you are using this module for your clinical trial application or for the IND application, it will trigger a change or update in the investigator's brochure, because everyone needs to know that there is a type of, I would say, there's a risk of carcinogenicity using your drug. Then for the reproductive and developmental toxicity, we will also expose animals to toxic doses, and then we are going to look into the effect of those high doses or toxic doses, single dose or multiple or repeated doses, on the fertility and early embryonic development. Because as you know very well, when you look into the what we call organogenesis, we know very well that when again all of our organs are not made at the same time, there is a type of trigger that we can see from just the conception of the egg, and then we will see that at the level of the gastrulation we are going to have movements of the exoderm through the I would say going right to the endoderm, and then this is going to cause differentiations of cells that will then continue as a cascade in order to generate different organs as different timelines. And again, of course, you can, I'm sure that you already learned about the organogenesis. This is where we start looking into, for example, creation of the neural tube, creation of the gastrointestinal system, creation of the eyes, of the heart. And what is interesting is that the central nervous system will still continue to develop until the age of 20, 25. Again, it depends on different studies, but this is why we are doing this type of studies. Now, obviously, there are some, I would say, complex discussions related to whether or not the results of the non-clinical data may affect the effect of the clinical data. But again, we need to go through the animal studies, and it will, of course, these are going to give us more information or some potential risk about our products, including, of course, the toxicity. Then we have to put prenatal and postnatal development and maternal function. This is for module 42353. And then looking into the offspring dosing studies and evaluations. We will also look into the local tolerance in Module 4236. For example, we're also going to look into the effect of the drug on the gastrointestinal system, the effect of the drug on a central nervous system, the effect of the drug in body systems that are not directly involved in our study, because we are looking into the effect of the drug, of course, in animal studies, but for its safety and efficacy to be used as a therapeutic tool for, again, resolving the signs and symptoms of a disease. So here we are looking into other side effects, but effects of the drug on other systems, not just a specific system that, again, is involved in our primary pharmacology. And then we can also add other toxicity studies such as antigenicity, immunogenicity, mechanistic studies, dependence to the drug, metabolites, other metabolites of the drug, because don't forget that a drug can be metabolized, but the metabolite can also be active. The metabolite can even be more active. I can give you the example of venlafaxine or Effexor, where when venlafaxine is being metabolized, one of the metabolites, because a drug will be metabolized into several molecules, not just one. And then the metabolite of venlafaxine is called desvenlafaxine, that has almost the same potency as venlafaxine. However, it has less adverse drug reactions related to the use of that molecule. Hence, and of course, I'm sure that now you understand that why, again, the company that did put Effexor on the market made another drug using the metabolite of that drug, so desvenlafaxine, in order, of course, to give a safer product to the population. And of course, we can also look into the impurity studies. And at the end, in Module 4.3, you will have to add the literature reference, the list of references that you use. So this is, in a nutshell, the table of contents for Module 4, which is, again, non-clinical studies. Now let's go into detail and look into, for example, a few of these documents. Okay, so one of them is the primary pharmacodynamics. So here, for the primary pharmacodynamics, we can see that, again, it's just here. It can, of course, be only, again, the summary of all of the primary pharmacodynamics. Don't forget that we are having, of course, here several references that we are going to writing here. If we do have animal studies that we did ourselves, in this case we will have to add those study reports in this again in this module. So you see here we are having, again, a type of summary, but again, it's not just a summary. The summary should go to Module 2. So here, this information is much more in depth. And this is, as I told you, as per all of the studies that we already conducted or performed for the primary pharmacology. So here for this drug, again, this is, again, another just a 6-page document about the primary pharmacodynamics, of course. Of course, if you are using any type of illustration from different peer-reviewed journals, you can always add those here. Don't forget that when we are also again providing the literature and the references, we also need to provide regulatory authorities with a copy of the again the literature that we have to add into our submission. And don't forget that those copies are copyrighted, and we will have to pay for being able to have or add one of those articles in our submission. And of course, it will continue until, as you see, we are done with the primary pharmacology, and then we are going to switch to secondary pharmacodynamics if applicable. Now let's go over, I would say, 4211, for example. Again, I don't open them in a chronological order. So here, this is about the, okay, this is for Sativex, for example. This is about the drug interactions. So drug interactions, as you know, Sativex is a cannabinoid, and again, this is also a drug as well. This is a medical marijuana that is being used, and of course, this is approved. So it's legal. Then I can talk about the absorption. So this is again information about the absorption of the drug. Again, don't forget that, as you see here, the number of the literature references can be up to 277 articles, sometimes more. So we will need to write this information and of course refer to all of the peer-reviewed articles that we will also add into module 4.3, which is for literature reference. So you also understand very well that writing this type of information needs some skills in medical writing, and of course here we are talking about regulatory writing. So then we can also talk about the distribution of the drug. This is again another information. Okay, that's about Sativex as well, which is a cannabinoid. Now the distribution is very interesting because we do have different compartment models, and a drug can be distributed again in different organs. It will depend which drug. It will depend also on here of course on animal species as well. What I can tell you is that once we have animals, again we have the studies, we can again order a biopsy in the animals, and then we can have up to 400 different types of tissues that we will be able to look at when we are looking into the distribution of our drug. And as you see, here we are looking into animal studies. So if you are having several animal species, obviously we are going to have several types of information. And you also see very well that now the number of the literature, again, references is going up to 300. Again, I can tell you that it's even more than 300, and this is again a type of, I don't want to say a thesis, but it's very similar to writing a thesis about a drug. Then our document is about the metabolization. As you know, we have metabolization that takes place in different tissues. Liver is the first and largest, I would say, it's not the first, but the largest organ of metabolization, where we'll be able to look into two types of metabolization. One of them is to break down the molecule into smaller molecules. And since we do have several metabolization enzymes, we will see the breakdown of our drug into not just one molecule or metabolite, but into several metabolites. It happens that we may not be able to track our original drug in the blood because it has been fully metabolized. Hence, we will need to learn about the different metabolites that we can track. And in this case, we call those metabolites biomarkers. These meaning that these are markers allowing us to follow where the drug goes and where the drug is going to be accumulated, where it's going to again do its effect as well. And by even learning about the distribution of the drug and metabolization rate of the drug, we can also get much more information about the safety of the drug. That's because you do have drugs that are again metabolized, that is, they are broken down and they are going to find themselves in the urine, for example, or in the blood as well, in order to be metabolized again. But we also have metabolization by conjugation, meaning that we are having, this is a second type of metabolization, where the body in hepatocytes, we are going to see molecules that have been added, again, another group of atoms, so that we are going to have the molecule that is not anymore the same because it also has other groups of atoms added into its structure. In this case, those conjugated or sulfonated also, of course, molecules that are metabolized, they are going to be too big in order to be released or even to be able to diffuse out to the blood. So what happens with them is that we will see those big molecules going back to the again to the intestine through the biliary tract, and then they are going to be excreted through the feces. We also have some metabolites that become gaseous, and then we will be able to find them in the exhaled air. We can also find again metabolites that are they may be active or inactive, it will depend on the drug. We can find them in the saliva and in the mother milk. This is why when we look into the I would say the effect of the metabolization, this information gives us more information about the potential safety issues of the drug. Don't forget that, and again, when you're going to work or read the reports from clinical trials, so trials in humans, you're going to see that in several protocols, we are going to say, for example, that people who are in the study, they should not give milk to their kids, of course, if they are if they are lactating. So this is because of the fact that we want to I would say preserve the well-being of toddlers who are taking the milk from their mother if their mother is in a clinical trial. So the metabolism again report is extremely important as well. Again, I may have to tell you that even the metabolization may not be exactly the same. The biomarkers may not be the same in animals compared in humans, but some cases they are very similar. This is why we still have to go through the animal studies. So about the excretion, I already covered that as well. What is interesting again is that the metabolites, as they may be inactive or active, they may affect, of course, the environment and, of course, the partners or, I would say, infants or kids of our subjects. What is also interesting is that even a metabolite, when it's excreted, if it is excreted, because if a metabolite is only liposoluble, for example, it has a very big, big molecule, and it may not even leave the blood, it may also accumulate into the body. So the study of excretion of the drug is important because we would like to know how much or what dose we added into or we gave to the animal, and if the exact complete dose has been excreted. If not, it shows that the drug is being accumulated in the animal. And again, here it will need, of course, more research to understand if there will be some toxicity if using a drug for a longer period of time. So another document that we can talk about is about the pharmacokinetics. This means what the body does to the drug. So here we are talking about the pharmacokinetic and pharmacodynamic relationship for, the document 4226, pharmacokinetics. So here we are talking about, of course, here you can see this is about the THC. So this is why here we are talking specifically about the smoking of legal cannabis with THC. And of course, we talk about the oral or oromucosal cannabinoids as well. So again, it will depend on your drug, its molecule, its stability, and of course, its use or recommended use. So here again, you are going to need to look into, again, that type of information and to, of course, add it into your module 4226. Then we also do studies on the toxicity of the molecule. So here we are looking into a simple dose toxicity. The single dose toxicity is when we just inject or we give one dose of a drug, and now that dose is more than, again, the safe dose range. So this is going to be toxic, and it's going to give us information about what are the signs and symptoms of toxicity for this drug in a specific animal species. Don't forget again that even if a drug shows some toxicity in animals, it may not show necessarily any toxicity in humans for the same dose. And of course, the opposite is also applicable, where if a drug is showing to be safe at the toxic doses in animals, it doesn't mean that it is safe in humans. So we have also to consider that as well. And here I can give you the example of thalidomide that was safe in rabbits, also in again in offsprings of the rabbits. However, in humans that was a catastrophe. Even if now, of course, nowadays we are using thalidomide, but under the very strict limitations, and this is only for cancer. So I also talk about the single dose, and here I'm just showing you as the studies. This is a tabulated summary of the repeated dose toxicity, and this is again for delta 9 tetrahydrocannabinol and cannabidiol, which is the Sativex. But again, for your drug, it may be different. Don't forget that in this document you will need to add a summary as well of the information, but you also need to have a narrative. So you have to add, again, here we don't have it, but in the original submission, we do put all of the study reports. So if you're now having several of these reports, we are going to add these study reports. If they are not done in the company, we will need to refer to different studies, and those studies must be added into the section 4.3 or the module 4.3, where we are going to add the original or the original publications in those in that module. Then we also have studies about the genotoxicity, where we are looking into the influence of the drug on the genes, and if this influence is going to cause any genotoxicity, meaning that at any specific dose, if those drugs may affect the structure of the genes that are, of course, again, very important to know about. And again, don't forget that if we observe any effect in animals, it doesn't mean that necessarily we will have those effects in humans. However, we can also see very similar effect, even identical effects of a drug in animals and humans as well. So these studies, these animal studies, are extremely important in order to know, as at least to know exactly in the clinical trials what we are going to look at. Are we going to look at all of the system organ classes that are about 56? Are we going to look into all of the different tissues that are more than 400 in our bodies? Or are we going to focus on observing specific signs and symptoms based on animal studies? So animal studies are going to allow us to, I would say, to fine-tune the limitation of our research, even if it doesn't mean that we will be able to cover all of the different adverse drug reactions, for example, related to use of the drug. And this is why when you are on the market, even when you are doing clinical trials, you will need to do pharmacovigilance activities, meaning that you will need to collect, assess, report, and prevent harm to, again, or adverse drug reactions to your study subjects. So through these different studies in animals, we will be able to look, to know in advance what are the signs and symptoms that we may see. However, If we miss those, and it happened to, for example, Vioxx, it's not just Vioxx, there are so many, again, drugs every year where we may have missed to look into the right, or to look into all of the potential adverse drug reactions that may be fatal or harmful. And of course, what we do now is that governments, for years now, governments, they allow you to go to the market as long as you're in compliance with the regulations, as long as you can show the safety and efficacy of your drug within your clinical trials, but within the framework of your clinical trials. Because when you go to the market, you're going to deal with people who are taking other drugs. They have comorbidities, they have other uses, they may misuse your drug as well. And in this case, we have to monitor that, get the information, update the monograph, retrain doctors, patients through websites, educational materials, and then to monitor the safety of your drug, not only again during the clinical trials but also on the market. This is why we also need to have pharmacovigilance activities during the clinical trials, as well as long as we are on the market. Now, talking about carcinogenicity studies. So here again, there are several carcinogenicity studies. We have to again add the again the information that we have. If we have any study reports, we have to add those study reports 1, 2, 3, 4, 5 in this module 4234, and then we also add this information that is going to embed all of the information. Don't forget that if you're having too many pages, you will need to add bookmarks. It's usually more than 5 to 10 pages that we add bookmarks into our PDF documents. Then we have studies about the fertility. So here we are going to look into the effect of again the drug that we have, or the active pharmaceutical ingredient. And here, as you can see, this is written in a tabulated summary. And again, we refer, of course, to the literature, and all of these articles that are now here we are again related to are going to be added into the section or module 4.3 for the literature. We also look into fetal development and birth defects as well. We also look into the offsprings as well. So here, these are studies that have been done in the previous years. We refer to them as well. If we don't have enough studies, we will need, of course, to do the other studies or new studies in animals as well. And this is where the department or the group for the preclinical studies is going to be very active as well. Don't forget that all of these decisions are made based on the research questions and research hypotheses, and we are doing all of these studies because we are focusing on the use of again your drug, your active pharmaceutical ingredient, for a specific disease, for these specific signs and symptoms. This is why we have primary pharmacology and secondary pharmacology. Okay, we cannot cover everything. We focus on one research question, and we have, of course, now in the studies we have primary outcomes, secondary outcomes, not only in the protocols for clinical research but also in the protocols for animal research. Don't forget also that for the ethics we have again ethics board for clinical trials, but we also have veterinary ethics board for conducting animal research. Then when we look into the local tolerance, this is where we are looking into again the effect of the drug on, for example, other systems like in a gastrointestinal system. So now we refer to studies that we have done by the company, or any study that has been done outside of our company but on the same drug as well. This is why the review of literature is a primordial task, a primordial job when we are preparing our submissions. And of course here I'm talking about module 4236, where we are going to look into existing information. So again, as I told you already, this type of writing needs again regulatory writers and medical writers and this is very similar to a thesis, but a thesis which is, again, very, like any thesis, which is very, again, focused on, focused, I would say simply focused on the efficacy and safety of the drug for a specific sign and symptoms. And of course, a specific therapeutic use. Now, for the section 42377, we can look into other studies in animal studies. So here we are adding, of course, for example, here they are talking about the mild doses of the drug, the moderate dose of the drug, and then they are looking into the severe, or say very high dose of the drug. And even if those studies may not be necessarily related to the primary outcomes or our research hypothesis, this is always an additional or supplemental information that maybe become, it is always important to know that, but they may also be necessary later. So we add them as well into our module 4. And of course, when you're looking at a literature reference, this is one way to write the literature references, but don't forget that usually we prefer, and it depends on the SOPs for each company, but we prefer to add a format where it goes beyond only writing the hyperlinks or the URL of the publication. So again, you have hundreds of different formats. In the academia, we are always using APA format, of course, for the references, but what I always encourage, again, my employees or my colleagues as well, and as my students, is to, of course, choose one way of writing that for writing references, and I always use personally APA, but again, it will depend on the company in which you are working, and it will depend on the SOP on how to write the literature references. So we looked into module 4 for the preparation of a common critical document. We looked into the table of contents. We explained each and every document that we have to put into the again module 4. We also looked into the section of module 421 for the pharmacology. We looked into the primary pharmacodynamics. We looked into secondary pharmacodynamics. We looked into the safety pharmacodynamics, and then we looked into the pharmacodynamics and drug interaction documents. We also looked into the pharmacokinetics, where we looked into the analytical methods and validations. We looked into the documents about the drug absorption, distribution, metabolization, excretion, pharmacokinetics and drug interactions, and other pharmacokinetic studies. And of course, we ended up talking about the toxicology. So we used about the single dose toxicology, repeated dose toxicologies, genotoxicities, carcinogenicities, reproductive and developmental toxicology, local tolerance, and of course any supplemental or adult toxicology studies. And we ended up talking about the literature review, and I talked to you about the fact that we always need to have a format, a specific and consistent format, such as APA format for writing our literature reference. So I just want to thank you so much again for joining me for this lecture, and I wish you all a great day. Thank you.